Thank you very much, and thank you for organizing this wonderful symposium. So within the context of white matter plasticity, if we want to extend this process in the brain, we can think of the connectome, the connectivity of the brain, and to see how this effect that happens on the myelin level affect the overall connectivity of the brain. So to put it in perspective, what is the connectome? So the connectome would be the mass array of all neural, not only neuronal, all neural connections that we have in the brain. Obviously, this is something that will be very difficult to measure entirely, obviously in the human brain. But we do know what are the specific components that contribute to this. Obviously, synapses and things that happen in the synapses contribute to the integration of the connectome, dendrites, dendritic tree. But importantly, in the sense of the white matter, it is the axons, it is the myelin and the glial cells that put everything together and contribute massively to the structural connectome at the end. And these are the structure, and it's not something that was newly invented. We use the word connectome for the last decade, but as we understand from the history of neuroscience, this concept was well established years ago by the drawings of Cajal, which actually shows the connectome and other methods throughout the years. All right. As pointed out by Heidi, so myelin is now well established as a component that undergo plasticity and undergo rapid changes, much more rapid than we thought. And it is not a stable component of the brain. That was shown very nicely on the molecular level. So we are starting in this talk from molecules and mice, but I promise you that through evolution we'll get to the end to the human brain. But in that work that came out from the lab of Douglas Field, he was able to show that by a very controlled cellular assay, he was able to show that continuous activation of neurons produces myelin on glial cells that are isolated in another chamber, which shows that neural activity actually promotes myelin within mere hours or days. Other work has showed in mice that repeated activation through optogenetic repeated activation to the cortex causes changes in the myelin content of the white matter below it, just as Heidi showed through imaging. So this seems to be that even though FA is not the best marker, it actually does show changes that are related to myelin in that. And also the seminal work by the group of Bill Richardson that showed that abnormal production of myelin may lead to abnormal plasticity at the end. But when we get to neuroimaging, we have to use diffusion imaging, because when we want to do in vivo imaging, or where we want to go to the human brain, at the end we have to use this marker. And regardless of the criticism that we have on diffusion imaging, which is rightly so, diffusion imaging is not optimal in many senses. There are many sources of artifacts and problems with diffusion imaging, and specifically with tractography, which is the measure that enables us to capture some properties of this connectome, the connectivity between different regions, despite the false positive and the false negative that we have in this method that was nicely highlighted in the last few years. I think, and I want to show you, and I hope you will agree with me, that there are some substantial information in this method that is true at the end. So, as was shown, so just putting white matter plasticity with diffusion imaging, so that work on juggling and the later work that was by Heidi's group that showed that in the adjacent gray matter changes you have a path of white matter changes that have changes in their FA. Also, we have shown that through water maze, spatial navigation, you see changes in the hippocampus and related area, but also some changes that happen in the white matter, for example, here in the corpus callosum. And also, with the running wheel, we use a different type of mice to look at this task. So in these mice, they have a problem in maintaining the myelin. So they can produce new oligodendrocyte, but when stress comes out, like in a very complex task, like the complex wheel, these mice will fail. And what we were able to show is that there is when these mice get to the complex wheel task, there is a marked reduction in the FA, which means that they can't produce new myelin. It's not only that they can't produce it, they actually, the oligodendrocytes just undergo some kind of cell death at this area. So altogether, diffusion imaging seems to be sensitive enough to capture some information related to myelin or white matter plasticity. But the question is, what is going on the overall network and overall connectivity? And specifically, there is a repeated observation of changes that happen on the corpus callosum, and there are several works that have shown that. And the question that we may ask: what is the role of the corpus callosum in general in the brain, and specifically in plasticity? So most obviously, is to transfer information between the two hemispheres. This is, I think, the most obvious role that we can give the corpus callosum. It is as obvious as drinking good espresso in Rome, to say that. But it must have some other more distinct properties or roles in the brain. So we do know that migration of cells after trauma, after neurodegeneration, happens along the corpus callosum. So, for example, with MRI or with other microscopy methods, it was shown that cells migrate through these large white matter fascicles on the corpus callosum to reach the area of lesion. But the question that we ask, what is the role of the corpus callosum? The corpus callosum specifically in brain plasticity, epigenetic, and efficiency of brain information transfer in general. And the reason that we ask that question is there is a growing amount of information in the literature that the corpus callosum has a pivotal or critical role in plasticity. For example, this work that was published almost a decade ago showed that mice that are grown in an enriched environment has a bigger corpus callosum. For some reason. And another work that in humans showed that there are various microstructural changes in diffusivity and FA in humans, in elderly humans, that undergo some kind of cognitive training and memory training. And there is a series of, studies that are focused on music, for instance, that show that, that the amount of time that we are engaged with training with music or playing an instrument or our ability to play the instrument is good is highly correlated in some section of the corpus callosum. And when you look at this kind of studies, the question that comes out is why? Why the corpus callosum should be such distinctly correlated with these various cognitive processes. And well, it is the major white matter system that we have in the brain, passing information through both hemispheres. And we may think that since it's the largest and most evident structure in the brain, here we see the 5 commissure, the corpus callosum in the human brain is the largest. It should be critical to that, but we must remember that it wasn't always like that. Because if we look at the kangaroo brain, for example, it doesn't have a corpus callosum, doesn't have a corpus callosum at all. We see a huge anterior commissure, we see the hippocampal commissure, but there is nothing on top of that. On top of that is only the cingulum. So the corpus callosum is absent. Obviously, the corpus callosum was evolved in a sense, and maybe this is why it is so critical. To explore that more in depth, we were lucky enough to get a database, to acquire a database of more than 120 different mammalian species, altogether 230 different animals, including almost every order you can think in the mammalian class. And just to note that no animals were deliberately harmed for this project. They just do die occasionally in zoo and in the wildlife. So we were able to get diffusion images and to explore the diffusion properties and the connectivity through tectography of all these different animals, just in order to understand how the commissural connectivity has evolved, and does it have a really significant role in plasticity. What we were able to see is that there is indeed some sort of evolutional process. So here we can see on the kangaroo, we see the anterior commissure and the hippocampal commissure. Through evolution, this is bad, this is the hedgehog, a rabbit, a wolf, you can see that the anterior commissure becomes smaller and smaller and smaller until it almost disappears in the primate brain, while the corpus callosum just evolves. It's not existent here. At the end, you have only the genu, then the splenium, then the body comes out. So it seems like an evolutional process. If we put it on the radial plot, so the percentage of fiber that passes through the corpus callosum, the anterior commissure and hippocampal commissure, you see that kind of evolutional process from the marsupials until the primates. So there is an increase in the amount of the corpus callosum. The problem is that this graph doesn't follow the evolutional tree. For example, the hyrax came, which was just diverged from the marsupial before all other animals, has already an evolved corpus callosum, and vice versa. The bats that diverged only later on doesn't have a big corpus callosum at the end. And it's not only that it's through evolution. We can look also in a specific order, like the bats. You can see that some of the bats, for example, in our database, we had about 20-something bats. Half of them have a big anterior commissure, and half of them have a big corpus callosum, which seems that within a species or within an order, you have this kind of variability. And we also can see that within the primates, for example, for the lemurs or the tamarins, they have a bigger, all primates have a big corpus callosum, but you see that some has bigger than the others on that end. So the question here of the evolution of the corpus callosum or the role of the corpus callosum is actually the most ancient question that we have, I think, in neuroscience, the relation in the brain, the relation of changes in the brain in relation to the nature, to the genetics, evolution, or the nurture, the epigenetic and plasticity. And it's very difficult to determine, based on this observation, what is, why the structure of the corpus callosum is that critical. And you can see that in these two examples, so this is a cave nectar bat and this is a fish-eating bat, only 50,000 years separate between them. One of them has a corpus callosum, one of them doesn't. And this is quite intriguing to answer that. In the analysis that we have done on this database, we have looked on the amount of information that passes through the hemispheres, the commissural ratio, how many fibers compared to the entire brain volume passes through the mid-sagittal lines, and compared it with the hemispherical mean short path. So mean short path is the index that you get from the graph theory. It's a sort of, without getting into details, it's a sort of a marker of the efficiency of the network. And what was interesting to see that animals or species that have a low amount of commissural fiber passing through had a very efficient hemisphere, and vice versa. Species that have very large amount of fiber passing through the mid-sagittal line had a very inefficient hemisphere to that end. And that also goes within an orders or the different orders in the mammalian class, where you can see that within primates you see this correlation, within rodents, within the bats, the carnivores, etc. So it's something that happens through evolution. There is an evolution process of developing the corpus callosum, but it seems to be highly dynamic even within an order, and actually even within a species, because we all know that so all of us are neuroimagers, when you acquire a human brain and you look at the mid-sagittal plane, it doesn't look the same for all of us. Some of us has very thin corpus callosum, some of us has very thick corpus callosum. And we do know that some of this is actually me. So with 15% of corpus callosum, I have, well, an efficient hemisphere, lucky for me, and vice versa for other people that have very thick corpus callosum, but their hemisphere or the connectivity within the hemisphere will be very poor. So there seems to be some kind of a dynamic process that the corpus callosum needs to maintain or to conserve. So that kind of relation shows conservation with information transfer in the brain. And if we undergo some sort of plasticity, it is no wonder that we see changes within the corpus callosum. And we do know that from people that have agenesis of the corpus callosum, this was a study that was published by Owen et al. In Neuroimage, when they don't have a corpus callosum because of a mutation, they too have very strong connectivity within the hemisphere. So plasticity of the corpus callosum can happen through development, through evolution, but obviously through plasticity. So to summarize, and this is the thing that I wanted to argue, is that the corpus callosum is not there just to transfer information between the two hemispheres, but to conserve the amount of connectivity and to balance off. It's not that when you do more and more and more practice, eventually the brain will explode. Something needs to preserve, something needs to make balance, and I think it's the corpus callosum. So with that, I will finish and will thank my collaborator and people who've done the work, and also the animals that fully contributed their brains to the database. Thank you.